Give a big round of applause for our today's eminent guest. A very warm good morning to one and all present over here, esteemed guests, dignitaries, and delegates. Welcome to the second day of International Conference on Approach to Pharmaceutical Insight through Interdisciplinary Research 2024. I am Shivam Yadav from 7th SEM School of Pharmacy.
And I am Kainat from 3rd SEM School of Pharmacy, GH Raisoni University, Saikera. And we are delighted to be your host for the second day of this grand international conference. We hope you had an inspiring and enriching first day at the conference. It's a pleasure to welcome you back as we continue this journey of knowledge and collaboration.
We are delighted to see so many eager faces today. We assure you today's session will be just as captivity as yesterday. To being, let's seek blessing of Ma Saraswati by lighting the lamp and offering Vandana. I request all the dignitaries on the dais kindly do the honor. Thank you to all the dignitaries. Insightful present- We witness insightful presentation and engaging panel discussion that set the stage for productive dialogues.
Absolutely. And today, we are privileged to host two renowned speakers who will share their expertise. Stay tuned for thought provoking content that will surely inspire.
I request Dr. DC Sahu to welcome Dr. Rahul Lad by presenting a sapling as a token of love and gratitude. Now, I invite Dr. Manisha Kavatkar to welcome Dr. Jitendra Madan by presenting a sapling as a token of love and gratitude. As we proceed further, I request Dr. Jignesh Doshi, Director, CIRD, GHRU Saikheda, to express his opening remark of today's scientific session.
A very good morning to all the dignitaries on the dais and off the dais, delegates, faculty member, and dear students. I welcome you all to the second day of the International Conference, Approach to Pharmaceutical Insight through Interdisciplinary Research 2024. Pharmaceutical research as a domain is highly interdisciplinary in nature. The journey begins with molecular biologists identifying a gene or a protein responsible for causing a disease, followed by efforts from medicinal chemists to identify small molecules or peptides to target this protein or gene and to cure the disease. This is, of course, done in coordination with pharmacologists who develop various in vitro and in vivo assays. This is then followed by pre-formulation and formulation scientists from the pharmaceutics domain to formulate the molecule in a form which is easy to administer or deliver to the patient, right? And the drug release profile and pharmacokinetics are studied by biopharmaceutics specialists. The data from all these preclinical studies is then compiled into a dossier, as Sir was mentioning yesterday, right? Which is prepared by scientific writers. And this is then applied for permission for clinical trials. Following the clinical trials, involving volunteers, nurses, doctors, statisticians, bioanalytical scientists, and of course, pharmacists, a new molecule is finally presented to the world. A similar message about interdisciplinary research was echoed by many of our speakers yesterday, as you might have heard. We had a wonderful lineup of speakers yesterday, sir, including our chief guest, Dr. Dipender Singh, who emphasized on community pharmacy and the role of pharmacist as a counselor for patients. The session by Dr. Shivam Srivastav from Zydus Group, Tanzania, highlighted the development of biosimilars and the career potential that is there in this upcoming field. In the post-lunch session, Dr. Manoj Tatwadi of Gau Vigyan Anusandhan Kendra, he highlighted the potential of traditional Indian remedies based on Panchagavya. This was followed by an equally impressive session by Dr. Abhijit Munshi of Alchemy Clinical Research Services, who elaborated on the imminent demand and the career prospects in clinical research field. I'm sure we'll continue this trend today as we have some brilliant speakers lined up for today. So without taking more time, I would like to hand over the mic and I wish you all another fulfilling day of great learning ahead. Thank you.
Thank you, sir, for your inspiring words.
Without further ado, let's introduce our first distinguished speaker of the day, Dr. Rahul Lad. Dr. Rahul Lad has done his MTech from Naipur, Mohali, India. He earned his PhD in medicinal chemistry from University of Minnesota, USA. He is having over 10 years of experience in healthcare industry, primarily in medical communications and medical affairs. Currently working at Novartis, Hyderabad, he handles high value, high impact projects including publications, patient engagement activities, scientific engagements, continuous improvement initiatives, etc. At global level in cardiovascular, renal, metabolism, oncology, COVID 19, etc. He has managed a large team of scientific writers operationally as team lead. Before joining Novartis, he worked as a safety data manager at Cognizant Technology Solutions India. Ladies and gentlemen, please join us in welcoming Dr. Rahul Lal.
And this works? Yeah, this works. Podium also works. Yeah.
I think we are good, right? All right, good. Thank you so much, first of all. Thanks to the organizers, the esteemed faculty at this institute. Thanks to all of you for taking time from your busy academic schedule. I essentially come from an industry background at this point, but obviously I was a student at one stage in my life, and I always aspired to be in the place that I am right now. So it's very, very honored situation for me. It's a pleasure for me, and thanks for the invite from Dr. Doshi and the faculty. Before we dive into the topic of my presentation, I think it's important to understand that the landscape of pharmacy is changing. We definitely want to focus on the fundamentals of pharmacy, pharmaceutical sciences. But if you look at the industrial landscape, the importance is growing for applied knowledge. By that, I mean whatever you learn in the classrooms, in the labs, it's important that you are able to apply those learnings, those fundamentals to things like clinical trial research or medical communication. In the next 1 hour, I'll try to be as interactive as possible, so please respond to me when I ask questions. So my first question is, how many of you have heard about medical communications before? Can you give me a raise of hand if you have? No, that's a good one. That's a good one. So I think we definitely have a knowledge need here. How many of you have heard of medical writing before? I think quite a few. How many of you have worked on a publication or read a publication from a journal? Have you read a publication, article from a journal? Yeah, good. Good. So I think we are connecting dots here. Okay, and obviously there is a need to learn more. So I think it makes sense to have this session about medical communications. All right. Good. So we can go to the next slide. This is a standard disclaimer that my company asked me to give. Even though I'm a scientific director at Novartis, I'm not an exception to this. So just remember that whatever I say today are my personal views, not of Novartis. Okay. Go to the next one, please. Yeah, so I always like to talk about the bigger picture, and this is what we all have to think in life as well. Even if we are doing small things, I think we always need to think of what bigger lies behind those small things. If you're doing a small experiment in the lab, if you're running a chemical synthesis in the lab, it might very well turn into a drug tomorrow. So that's the bigger picture. So I always like to start my talks with the bigger picture. So let's understand the context behind medical communications. Why is it important? Why is it a topic of discussion? So if you can go to the next one. So we have to really think, and this group is very well placed because we are pharmacy, academia, we are students, we are faculty, we are the future of pharmacy. So we have to understand where we are going in terms of where the medicinal practice is going, what doctors are doing. If you go back many years, in the 1960s, 70s, a few decades back, all the medicines and its practice was driven by doctors. A patient would fall sick, they would go to the doctor, and whatever doctor prescribes, they would just blindly accept it. You know, you give me a tablet, a colored tablet preferably, and I'll take it. I'll feel good. Over the years, what happened is, the research progressed, and we also started finding new methods of formulating medicines. So the formularies came into picture. Formularies, compendia came into picture, and they started guiding the practice of medicine. They started guiding or helping the doctors in prescribing medicines. So doctors were also not making informed decisions earlier, but they started making informed decisions based on the formularies. So formularies, compendia, all the guidelines started to take preference. Coming to the recent times, doctors realize that every patient is different. I cannot prescribe the same drug to the all my patients that I see, or even among my patients, people might react differently, or patients might react differently to the drugs. Some drugs may be effective in some patients, but some may not. So that's where we started looking into the precision medicine practice or personalized medicine practice, especially in the developing countries or developed countries. This has been rising. And now we are in the age of genetics. And everybody knows that the human genome has been mapped. Genetic medicines have been developed. You always hear in the news that there is this 16 crore rupees injection needed for a small baby, and people are crowdfunding for that. That's basically a genetic medicine. That's based on editing the DNA or editing the genes. And that basically helps those small babies to grow into adulthood. So that's revolutionary. And we are looking into such genetic medicines, DNA based medicines, very precision medicines, even in oncology, medicines that would simply target only the carcinogenic cells, not the normal cells, right? So that's where we are going. Now, why is it important as a context is that, and we all lived through the horrible pandemic years, right? Everybody was looking for information on the best drug against COVID-19. We all were on the internet, on the Google, we were on WhatsApp University. We were hearing from every friend, every relative, everybody that this medicine, this CADA works, this method works. So the importance is also on the information. Whatever is happening in terms of medicinal practice has to be communicated as well, and communicated in a transparent way, in an accurate way. That's why medical communication, research communication is very, very important. So I hope that gives you the bigger picture. If you can go to the next one. Talking about India, if I see this crowd also, I see potential. I see people running experiments in the labs, going into the field, also doing industrial pharmacy work, community pharmacy work. So India is very prolific. We have large numbers. We have the highest number of pharmacy professionals in the world, maybe. What these professionals are doing in academia and industry are they're doing experiments, they're doing clinical trials, they're running research, and they're getting a lot of output. But that output, unfortunately, is not getting out there in the world. We are still lagging behind as a country in terms of publishing our research, in terms of communicating our research to the world. If you look at the whole numbers, maybe US and China are way far ahead of India in terms of this. There is a lot of gap in communicating research. Engineering is still ahead of the curve. They're doing good. They're publishing a lot of their research, but there is a need that even the medicinal research, the pharmaceutical research has to be communicated and published. So we have a huge scope here as pharmacy individuals. If you can go to the next slide, please. And now we'll go to the basics, okay? Now we'll go to the basics. Back to basics. We have learned the bigger picture. We have talked about the huge things we can do, but ultimately communication is about storytelling. Every day we are telling stories to each other. Our faculty are telling stories about research to us. We are telling our stories to others. Others are telling stories. So storytelling is perennial to us. In the context of Indian culture, we have epics like Mahabharata or Ramayana, which are basically stories, which our ancestors formulated as stories, and we really like them. Those are very consuming to us, right? To the Gen Z, to the millennials, is the Marvel universe, is the DC universe that engages us because it's a great story. It's a great story about superheroes as survivors or like as saviors of the world, I would say. So storytelling is essential in any field and especially in research. If you can go to the next one. There is an art to it though. You must have heard lectures where you almost dozed off the next second. It's very natural. I have yawned a lot during a lot of lectures, during a lot of storytelling sessions. So it should be interesting. Storytelling, that's why it's an art. And it is all about sharing with others what has happened in an engaging manner. Why it is important? Because it connects people through shared experiences. When we are telling stories, we are basically sharing experience, sharing our research in our medical field. And to be a good storyteller, what we need a very good premise. In movies, they call it the script. The script has to be very solid. The premise has to be solid. There has to be strong characters. In terms of research articles, the drug has to be very good. The medicine has to be very good. The research methodology has to be very good. And a confined space and smooth flow should be there. So whenever we are telling stories, it should flow very smoothly. And to conclude, a good story is always a good story told, or in our case, a good story written. That's why it becomes a good story. Next, please. When it comes to telling stories about research, there are many benefits to communicating research. And a lot of us find different purpose in telling stories about our research or community stories. Like in academia, especially, I think it's important to publish articles regularly and in a significant volume to further our career prospects, to get tenure, to get promoted from an assistant to an associate professor, for example. It's very important that you publish continuously. Also, to secure funding. How would a funding agency like DBT know that your research is useful or prolific unless you publish it? So we need to publish our research in good scientific journals to secure funding. Obviously, it gives us a good recognition also. When you are known to the scientific community through publications, they will start respecting you. That they will start respecting you as a prolific publisher of research and a good researcher. And ultimately, when you create an evidence through publications or communication, you are creating your own intellectual property. You have your stamp on it. You will be an author probably. You will be an author, co-author, lead author on a publication. So it becomes your intellectual property and it gives precedence for others to do similar kind of research. And the ultimate goal of medical communication or research communication is to disseminate knowledge. Because if you remain in a silo, if you continue to do your research in your own lab and you don't communicate it to others, there is no use. So we have to disseminate knowledge so others gain that knowledge, the society as a whole progresses. Next one, please. Now let's come to the essentials of medical communication. I'll tell you some details about what medical communications is actually. To the next slide, please. To me, this was a case actually, and this happens regularly, who doesn't know medical communications and somebody asks them to prepare an abstract, they might end up in drawing an abstract painting. But I think this community knows very well what an abstract is. It's basically an abstract for a conference or a publication, right? So we don't want to be in that poor guy's place. We want to be more knowledgeable. That's why we are going to learn about medical communications. Please, next, please. And going back to history again, this is not a new art or new profession to, especially Indians. We have a very good, very strong precedence in medical communications. We have the Sushruta Samhita that many of us would know, which is a core document in medicine practice, and it was basically founded in 6th century BC, so very old times. From old times, we are communicating research and we are documenting it. You have Charak Samhita, which was actually formulated around 3rd or 2nd century BC. So very old science of medical communication. It's not new. It is just gaining more recognition now. And we have to realize the recognition. Can you go to the next slide, please? Now, as a pharmacy student or a faculty or a professional, is scientific writing or medical communication relevant to me? Everybody should ask that question. And these are the answers. Essentially, anybody who is doing research, medical communications is for them. If anybody likes to tell stories to others about their research, let it be family, let it be friends, let it be your peer students, peer faculty. If you like telling stories about your research, medical communications is for you. If you aspire to be a speaker, if you aspire to present your research to the worldwide healthcare community, medical communications is for you. If you would like to publish your research, gain recognition, gain funding, gain that intellectual property, medical communications is for you. So essentially, if any of these applies to you, medical communications is for you. Let's go to the next one. Now, what is scientific writing as a definition? Basically, it's not just a form of creative writing. We cannot just write in any way or method that we like. There is a method to it, there is a principle to it. So it is basically a form of written scientific communication, and which is well structured, clear, concise, and effective, and which is essentially written by a person who is knowledgeable about the field. So, pharmacy graduate can very well write a well structured scientific article in pharmacy, but probably not in natural arts or other commerce field, right? We cannot write that. So one has to be knowledgeable about that. The material that is produced should be in compliance with the guidelines, and we'll come to that later. And these guidelines can be prescribed by the regulatory authorities like FDA, journals, and conferences. So that is important to know. And also important to know that we should not see at medical communication as just a supplementary to research. Many times down the line, people like us realize that I should have published more. I should have communicated more. I should have gone to that conference. I should have, presented my research. So it is not an alternative to research. It's basically supplementary to the research. So when you do research, also aim to publish it or communicate it. Let's go to the next one, please. And communication, scientific writing leads to success. So I think this is, how you have the central dogma in biology, right? Everybody knows the central dogma, DNA to RNA to protein, right? That's how we do in industry, in clinical research, in pharmacy. We generate evidence in the lab, right? We conduct experiments, we get outputs, we generate evidence, which is basically your raw science. But something needs to happen to that, right? It cannot stay in the lab or in your research premises. It has to be disseminated. It has to be circulated for scientific recognition to your peers, your doctors, your patients, and media as well. And what it does is when you disseminate, when you communicate research, it also leads to education and training of our healthcare community and our patients, importantly. That's why medical communications or scientific writing is a formula for research success. Let's go to the next one, please. Now, traditionally, I think everybody would be aware of research articles, printed journals, or even the conference posters, abstracts, oral presentations. We all, I think, are very well aware of these. But in the current age of information and digital media, we are not limited to only the published or printed media. I think there are many avenues to scientific writing now, including journal publications, scientific presentations, events. When you happen to go to a conference, I think you will be amazed to see that besides posters and oral presentations, there are many other activities happening. Focus group chats or focus group connects, meetings between patients, doctors, researchers happen. So I think we can leverage communication for that. We can also put our research on websites. Let's not limit ourselves to printed journals only. Let's utilize websites as Medscape. Everybody might be aware of Medscape. So there are many websites available which are happy to accept your research communication and which will be very useful in communicating that. And social media, of course, now Twitter, or I think it's called X these days. So X, formerly known as Twitter, is not just social media to express our opinions about politics, sports, and other things. It has also become a media for research communication. Just try Googling any conference, any major conference name in your field, or just try putting the hash mark and pharmacy. You will see millions of tweets about research. People are actively using Twitter, X, and even other social media platforms to communicate about research to each other. So don't limit yourself to just journals, but think about all these possibilities of communicating your research. And I think it's important not only for students, but also for institutes to use such platforms, which are very active social media platforms. If you can go to the next one. Now let's look at the core aspect of medical communications, because it is still the most prolific one. Next one, please. Scientific publications. Scientific publications is a must for a researcher, I would say. And basically, these are permanent record of our research. Nobody can delete this record, especially if you publish in a peer-reviewed journal, index journal. By means of publication, as I previously said, we are educating people, we are advancing the science and research. And they're basically your passport. If you have like 15, 20 publications tomorrow, and if you're looking for a job or looking for opportunities in academia also, this is essentially your passport. People will look for how many number of publications you have done in your CV. That's a very critical part of your CV. So nothing to be ignorant of, is basically becomes your passport to gain entry into all these avenues, including industry. So next slide, please. And just to show you some numbers to realize what we are talking about in terms of volumes of publications, you will be amazed to see that each year 2.5 million or more publications happen in all the journals. 2.5 million, and the number is growing. The number of oncology related publications especially is growing a lot, because a lot of focus is on cancers, different types of cancer these days. So 300,000 publications were solely on oncology in 2021, and the projected growth is 36%. So one third of the current volume is going to be added to the number of publications. So it is a very, very important field that is continuing to grow. Let's go to the next one. And I think this group may be aware of the clinical trials, right? Phase 1, phase 2, phase 3, phase 4. So in a typical lab setup, I think publications happen when the research is concluded, like we have conclusive finding. But I think it is important to look beyond the laboratory research and look into the clinical aspect of our pharmaceutical research. Like somebody said earlier that, whatever we do in our lab is actually going to end up as a drug probably, and it is going to be used by the patients. And for that to happen, companies or academia have to run clinical trials. So in clinical trials, in every stage, there is a need to communicate research. It's not just that you run a big clinical trial and there is a big hoopla around it, everybody wants to know about it, and then you publish. It's not like that. You can publish at a very early stage, even at in silico modeling phase, or even at formulation phase, phase 1, early stage, pre-launch as we call it, before launching the drug, and even after the drug is launched in the market, drug is available. So there is always a need, because there is always new evidence generated, right? You generate evidence at every stage of clinical trial. And I think, I don't know how many of you are aware, but even after the drug is approved and used, there is evidence generation. These are called as observational trials or non-interventional trials. companies or academia will not give any new drug to the patient, but they will just observe. Like, for example, if aspirin was launched 5 years ago, the research will still continue to go on its safety aspects. How safe is aspirin? Because in a typical clinical trial, you will probably have thousands of patients, but when it goes to the market, the number of patients using it is in millions probably. So you get a lot of evidence. So the point to make here is there is always a need to communicate and publish research. Next one. Now these are the main types of publications. I think these are quite familiar probably. When it comes to Congress presentations, these are abstracts, posters, oral presentations. And when somebody wants to publish in journal, these are probably the primary manuscripts, review articles, original research articles, or some narrative reviews, and even some small articles. You know, like if you did an amazing research in the lab, but you cannot wait for New England Journal of Medicine to take 6 months to publish that research. So what will you do? You want this research to immediately go out into the healthcare community, so you will write a letter to editor, to prominent journal, which is a rapid communication. They will communicate, they will publish it rapidly, and your research will be out there, right? So that's another way of publishing. Go to the next one, please. And like this tells you the details of it. I'll not go into the details more, but how many of you have referred to a review article, for example? Can you show your hands if you have review articles? So it's quite normal to review to refer to a review article, right? Because what it does, it gives you the whole landscape in that therapeutic area, in the disease area, or the drug. So review articles are basically summaries of all the published research in that field. Whereas original research articles, which we call primary or secondary manuscripts, are the ones that are typically very focused on one particular medicine, one particular type of research, and they will be published in high impact journals. Next, please. Just snapshots of what you may have seen before as well. Abstracts, I think many of us would know, are very short summaries, very limited in word count, 200 words, probably 300 words. And basically, these are your gateway to get into a conference. Like if you want to speak at a conference, first the conference would probably ask you to submit an abstract, and then it is accepted, and then you can speak or present at the conference. And the presentation can be in the form of a poster or an oral presentation. One thing to remember, guys, because you know what I've seen is 10 years ago, posters and oral presentations used to be very text heavy. Bullets, 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 bullet points. Please refrain from using a lot of text. Use visuals. Nowadays, everybody's on their phone. Even now, while I'm speaking, many of us are on phone. So everybody likes their phone. And with the phone usage that has increased, our span of attention has also decreased. You cannot look at me for 5 minutes straight. I think if I talk for 1 hour, everybody will sleep. So it's probably not just about me, but for anyone. Like we are not, we are living in the moment these days. We are not ready to spend a lot of time. So when you are speaking or when you are presenting at a conference, people will not have time to stand at your poster for 1 hour or 30 minutes even. So try to make it visually engaging by using a lot of pictures. When you prepare your next poster or oral presentations, let it be devoid of any text. Use pictures, use animation, use visuals just to make it more engaging because the audience has changed. Let's go to the next one. And yeah, we saw this. I mean, I think this is just a more detailed description of an abstract. So basically, it is the basis of your presentation. If your abstract gets accepted, you get to present. If it gets rejected, you cannot go to a congress. So it's important to think about abstract writing. Next one, please. Posters like you see here, it's more visuals, more graphs than text. So I think try to follow this practice when you're preparing posters or oral presentations. Next one, please. Oral presentations are very short At least for posters, you can stand there for hours and you can have people coming in and they have ample time. But for oral presentations, they typically give less than 10 minutes. So you have to present all your research in that very short amount of time in a very effective manner. So it's important that you use a lot of visuals, graphics, animations. Important to notice also that this is also an age of QR code and scanning. So if you are preparing a poster tomorrow or an oral presentation tomorrow, some people may not make it to that meeting, or some people may not be able to attend that your session or that session. So if you can, always include a QR code on your poster. And it's very easy to do. I think there are many apps available where you can put a URL link to the QR code so that people can actually scan the QR code on your poster, on your oral presentation while it's being held, and then they can refer to it or read it later also. So it becomes more perpetual. So one more tip to give you: when you do your own presentation, try to use QR codes as well. Next one, please. Now, these are new ways of scientific writing. Again, expanding on what I said earlier, it's not limited to printed. You know, when you go to congresses also these days, you know, conferences also, you will see a lot of big screens. You will see a lot of TV monitors rather than posters. Booths also. Try to go to booths of companies. They will have media. They will have videos playing. They will have quizzes playing. They will have animation. So if a company wants to show their novel drug on oncology, they will probably have a animation video playing on the screen, and people can actually put their headphones on and then actually click on the play button. They will immerse into that experience or mechanism of action of that drug. So on the other side, if you are the one who is going to prepare such communication, try to think of these new ways. You know, try to use multimedia, and there are many softwares available. I think we are all tech savvy in this generation, so you will find ways to do that. Next, please. Now, important to remember, whenever we are doing publications in a novel way, it's fine, fantastic, engaging way, but we always have to stick to the principles and guidelines. There are two key guidelines here, especially related to publication of clinical research. And the ICMJE is quite well known. I think it applies to all kind of research publications. So ICMJE stands for International Committee of Medical Journal Editors, and they continue to update these guidelines. Basically, this ICMJ guideline is important to understand who qualifies as an author. I think it's a normal practice. I would not blame it on traditions, but I think it's a normal practice. Suppose in a lab, if there is a senior PhD student or a senior professor or senior faculty, they would end up reviewing a lot of communication, a lot of articles and manuscripts. So, as a thing of honor, we might be tempted to make them as an author. I think it's totally fine. I think they can be an author, but it is important to actually give credit to the right person as an author. So, an author, what this guideline says is that the author should own the research. I cannot make an individual an author from another university who does not even know my research. They should be able to own the research. If tomorrow somebody comes to them and asks about that research article, they should be able to explain that research to them. Second thing is, they should be the one who did the research. An author should be the one who actually did the research. Third one is, the individual must have seen the drafts of the manuscript while it was being developed. They must have at least seen or reviewed the drafts, if not written. And the last one is that whoever is going to become an author should approve the publication. They should stamp it that this is good to go. So all these criteria is prescribed in the ICMJ guideline. I would highly recommend, whenever you start writing an article on clinical research, please refer to ICMJ guidelines. The other one is GPP 2022. This is basically more for industry sponsored or company sponsored publications, just to ensure transparency, because we see a lot of cases where many companies would make false claims about their drug, and they would support that false claim with a publication, which is a false publication. In COVID-19 times, many publications came out from a certain country which were ultimately proven to be false. And at that time, people followed those publications and used them as guidance for clinical practice. So that is a very dangerous practice. And to avoid that, this GPP 2022 lays out some guidelines for companies, for industries to adhere to transparency principles and also to be accurate in their publishing. Next one, please. And again, to reiterate why we should adhere to guidelines, although we don't like to follow rules, but why should we adhere to guidelines is that, this is going to impact patients. Our research in pharmacy, our research in the labs, the industry is going to translate into medicines and it is going to impact patients, doctors. So it's very important to be transparent, to be adherent to the guidelines. Very important to be ethical, right? These guidelines ensure that we are ethical in reporting. Guidelines also establish a framework. We cannot just haphazardly write publications in any way that we like. So these guidelines help us in giving that framework to publications. And also importantly for a university or a college or industry to maintain its reputation. When we publish strong evidence adhering to the guidelines, we maintain that trust in public, we maintain our reputation. So it's important to adhere to the guidelines. I hope I'm on time. Next one, please. Now, the most important part, career prospects in medical communications. Next one, please. So I have spent more than a decade in medical communications while my research was on basic fundamental oncology. So it was a clear shift for me, for a student like me who would rather aspire to be an R&D scientist, but I ended up in Novartis as the medical communications director. So how can that happen? And all that I told you right now can make it happen because it is important, but it is important to understand what is medical communication in terms of career prospect. So when it comes to medical communications, the spectrum is quite huge. If you go to any research website or any WebMD, Medscape type website, you will see promotional material also. You see Glaxo ads, you see Pfizer, Novartis ads. Some of them are promotional also. They're very promotional, especially consumer health, Complan or Bonvita. Basically, they're promoting, but as pharmaceutical companies, we have a mix of it. We also want to promote our drugs, otherwise we would not be able to sell our drugs. So there is a mix of medical communication that happens, and whatever is promotional in nature, medico marketing as they call it, brochure, pamphlets, or these advertisements, these are very loosely regulated. There is no strong regulation on it. People can make any claim like your height will grow this much. They can say that. You will see those ads on TV. Like overnight you will get rid of your arthritis with this balm. So there is no regulation as such on that. And there are writers who are writing that content. So those are also medical communication professionals. They're very loosely regulated, but we don't want to become those people ideally. So if you can go to the next one, this is where we want to be. We want to be in a regulated space. We want to be in a space of journal publications, congress presentations. We want to be doing material on efficacy and safety of drugs. We want to be doing material that helps patients. So in medical communications, it's not just about doctors or trying to promote our drugs. It is also more about patients. So patients are increasingly getting more knowledgeable. And as patients ourselves, we would have realized that, right? I mean, we are very cognizant about what we are taking these days. We do not trust, and we need more information about drugs. So what big companies like Novartis, Pfizer, and GSK, so forth, they are doing is they are focusing more on patient communication also. They're making things very simpler. If tomorrow I say this PPR gamma receptor drug is very, very efficacious and tolerable in this subpopulation of Asian patients, patients will not understand that. But if you make it very simple, that this novel diabetic targeted drug is very effective in this subpopulation, people will be more understanding about that, they will consume it very well and very easily. So a lot of emphasis is on patient communication also, patient oriented brochures. So these days when a doctor will give you an oncology medicine, they will also give you a pamphlet with that, how to use it, how to carry it, how to store it in a very, very simple language. So that's where medical communication also comes into picture. Slide decks for trainings, we all do PowerPoints, right? But these PowerPoints are not just limited to education in a particular setting. Those can also be used by people who are on the field, people who talk to doctors, or even doctors who present. So that's an education media. So that's where medical communications as a scope of career comes into picture. A typical scientific writer or a medical communications individual will be doing all these things. Next slide. Now, if you are going to apply for a position in scientific writing, publication writing, or medical communication, medical writing, what do you need to have in your resume? I think it's important to know. Hmm? So, first of all, it's preferable to have a master's degree or an advanced degree in life sciences. Let it be pharmacy, medical, dental, homeopathy, and others. So, I think any advanced degree in health sciences or life sciences is preferable for a career in medical communications. Obviously, the individual should know about pharmaceutical research. In-depth knowledge of the guidelines that I just mentioned is important. People in interview will ask these questions to the candidates. Excellent writing skills are recommended. It's okay if you don't have those writing skills, you can learn, but it's recommended that you practice writing. You can start writing essays just on anything, but start practicing writing. Strong communication skills, be confident, speak in the meetings, ask questions when such sessions happen. Be verbal, be confident in your written and oral communication, both. Like we use a lot of emails these days, messaging these days. We don't talk to the person, we just write a message, right? So you can even try working on those messages, how you write. Self inspect those messages for editorial mistakes. Try looking at boards on the streets. You will find a lot of editorial mistakes on the boards, especially painter shops or tire puncture shops. So things like that. Try spotting those mistakes in the boards. Try spotting those words on the boards. Even while you're traveling, try looking at things. Try reading, writing things. And again, if it is something that is recommended, that's the experience in publishing also. I think you're doing research already, so focus on getting it published also. Try to get it published. It is important that you have that in your resume. Strong knowledge of digital tools. I think most of us might have it. I think there's not a question about that. Ability to multitask, especially when you go to industry, you will be doing not only writing, but you will do a lot of project management. There are processes, there are frameworks, so you will have to adhere to those processes. So a lot of project management, lot of functional work goes on. So you need to multitask at all times and also manage the timelines, because we will not be working on just one manuscript at one time. You might be working on multiple projects at one time. So how you manage time, how you actually complete the projects within those deadlines is very, very important. So that time management skill also is important. So core skills, I would say communication skills, strong research acumen, scientific acumen, and confidence in communicating and aiming to publish your research is what we are looking for. Next, please. Where do these scientific writers or medical communication professionals work? So you might see them in pharmaceutical companies like myself. A lot of multinationals and Indian companies have understood the need of medical communication. So in Hyderabad itself, we have offices of Novartis, Sanofi, Amgen, BMS, Pfizer, who have dedicated medical communication groups and large medical communication groups where there is a huge need for pharmacy graduates like you to come in and become those professionals. Large Indian companies like Dr. Reddy's, Sun Pharma, Cipla, Lupin have their own medical communication functions, dedicated medical communication functions. So you'll find these individuals there. You will have agencies also, because many times it is not possible for an industry or a company to get that writing done themselves, so they will give it to agencies. So there are a lot of creative writing agencies, medical communication agencies. Some of the names are Indigene, if you have heard of that. That's a big one. Cactus Communications is one of them. So those are some of the examples. Contract research organizations, CROs as we call them, they work very closely with the patients, with the sponsors, and they also need medical communication professionals. Obviously, journals, who are the users of your communication, they will also need reviewers, editors, and writers. And academia, of course. In academia also, big universities, big institutions have medical communications teams, because imagine an institute like Tata Institute of Fundamental Research, which does a lot of research in oncology. They have their own medical communications team. So you will find opportunities there as well. And then again, large research institutes like I just mentioned. Huh? So a lot of avenues available, lot of career opportunities available to all of you. So how to prepare for that on the next one? Before that, I'll just show you the need. Like we need, as an industry, as a healthcare industry, as a healthcare arena, we need scientific communication people, medical communication people. We need pharmacy graduates to become scientific writers, because it is estimated that this will grow by 7% in the next 4 to 5 years. And obviously, you will see a lot of research happening, a lot of new drugs are being launched. So there is a need to communicate, like I said. So that's why there is a huge potential for people like us to become medical professionals in the healthcare industry. Next one, please. How to prepare for this career. And again, I'm going to re-emphasize a few things here. Try to gain a strong academic and research expertise. Try to ace your exams. Try to do well in your academics. Try to be a good student. Try to understand the research. That's all basic, like fundamentals, nothing to reiterate about, but just to say that we should have that strong scientific acumen. Work on the language and communication skills, because in multinationals or even in Indian companies, you will end up talking to people from the whole world. You will have a global audience. Even if you want to publish a paper, you will write that paper for a global journal, probably, right? So you will need to work on language and communication skills. Communication includes both verbal and written communication. Contribute to scientific discussions. If your institute, your university has forums for scientific communication discussions or research discussions, journal clubs, try to be a part of that. Try to put your opinion. Let it be wrong, let it be right. It's nothing like that. But try to become part of those discussions. Contribute. Get trained in scientific writing if possible. If you go on the internet, there are many websites that offer professional courses in scientific writing, medical communications. Many companies sponsor that. Universities like Harvard, MIT, and even Caltech sponsor these kind of courses on scientific writing and medical communication. Join a scientific society, be it a local society or a regional society, country, global, let it be anything, but try to join and go to the conferences. Go to the conferences, see how people are presenting their research, see how they're talking about their research, and then you will gain the knowledge. And ultimately, again, aim to publish your research. Aim to publish it. I think there is a very good saying, publish or perish, right? Especially in academia, it's very important. You either publish or you perish. So it's important to publish so that you don't perish. Next one, please. And this is the cartoon that I like very much. It's basically about a paper being accepted at the end, but not without hurdles. Huh? There are many hurdles in getting your research communicated, getting it published. I'm not saying it is very easy, but regardless of those hurdles, try to communicate your research to the wider community and try to publish it. Thank you so much. I'll be happy to take questions if there's time left. Otherwise, about career, about the scope, especially.
I just had a quick one. Like we know, now we have Chat GPT and other tools in terms of writing content and all. But when it comes to scientific writing, analyzing of data. So if suppose I look at some information which I obtain from Chat GPT, it is not analyzed by human, right? So how do you see, is Chat GPT a threat to scientific writing? Do you see that it's a threat, or it's not? Like,
It's a very relevant question, and I'm sorry I missed that aspect of AI, that one has to be cognizant of AI capabilities also to prepare for medical communications. And when this AI boom started emerging in recent times, there was a huge fear in all kind of industries, especially in healthcare industry and medical communications, that this is going to take our jobs, because chat gpt or other AI tools can easily describe anything that you wanted to describe. Like just the other day, I asked chat gpt to write something about me, and it wrote like 10 lines of only praise about me, nothing bad, because it doesn't want to be bad to you. It knows that you are the user. So that's where the question comes, that it has to be reviewed. The output that is generated by chat gpt or other AI tools has to be reviewed by a human. So that's where the risk is mitigated. So there is no need to get afraid of AI. The important thing that is we become capable in using AI. You cannot sit in a silo in a corner and just to be afraid of the AI. Oh, it's going to come and take my job and I'm not going to do anything about it. So I need to get trained in AI as well as an individual. So that's important. The aspect of intuition and creativity is very unique to human beings. Human beings are the only creative animals on the planet. We are very intuitive, very creative, and we can very quickly adapt. AI will take its time to do that. AI, the information that you feed AI, it will take that as the truth or as the core. So we can play with the AI. So basically, try to own AI rather than AI owning you. So because of the review requirement and the intuition and creativity aspects of human being, I think there is no need to fear. But having said that, again, try to be, try to be conversant with AI, try to learn AI, and try to be familiar with AI. That's the way to go. Scientific society, I think there are many societies which offer registration for free. Many of them will charge a fee, but there is no hurdle in becoming a member. Huh? So I think the key here is to identify the society that relates well with your research. For example, if you are doing pharmaceutics research, try to identify a society in pharmaceutics field. Try to become a member of that society. Especially when you are a student, you get a lot of discounts and exemptions for membership fees. So this is the right time. Industry people don't get that, but I think when you're a student, this is the right time to join the society, and I think you can continue to be a member of that society for many years. Any other question? All good. Thank you. Thank you so much again for your time.
Thank you, sir, for that enlightening session. Your expertise has undoubtedly expanded our understanding. Now it's time to introduce our second guest speaker, Dr. Jitendra Madan. Dr. Jitendra Madan is an associate professor and head of the Department of Pharmaceutics at the National Institute of Pharmaceutical Education and Research, Naipur, Hyderabad. With a strong academic foundation, Dr. Jitendra Madan has made extensive contribution to pharmaceutical sciences, completing his B.Pharm at Nagpur University, M.Pharm at Dr. Hari Singh Gaur University, and PhD at Bundelkhand University. He also completed a postdoctoral fellowship at Georgia State University, USA. Dr. Jitendra Madan is an active researcher currently supervising 13 PhD students. Okay, let's put our hands together and hand over the dice to conduct the session.
Thank you very much for the nice introduction. And first of all, I would like to thank the management at Raisoni University and the faculty members who has invited me as a speaker. So my presentation is especially useful for that is the pharmaceutics faculty as well as pharmaceutics students, because topic of my presentation is lipid nanoparticles. So as sir has said that in academia either you have to publish or you have to perish. So basically my work is especially related to the publications as well as further scope of this publication. So my topic is lipid nanoparticles as a carrier for topical delivery of meloxicam in knee osteoarthritis. Next slide, please. Next slide, please. Okay. So lipid nanoparticles are basically composed of the lipids which are solid at room temperature. Basically, they are the solid lipids. Okay. So these are the lipids which are solid at room temperature. So we can explore these lipid nanoparticles for delivery of both hydrophilic as well as lipophilic molecules. And in this publication or in this research work, we have explored these lipid nanoparticles for the topical delivery of COX-2 inhibitor, that is the meloxicam. Currently, there is no topical dose form of meloxicam is available in market. So meloxicam is especially useful for the osteoarthritis both in the animals and the companion animals as well as in the humans. But in China and Russia, topical formulation of meloxicam is available, but in India still it is not available in the market. Okay. So next slide, please. So these are the topical applications of the lipid nanoparticles. So we can improve the skin permission efficiency of any drug molecule using the lipid nanoparticles, and we can offer controlled and sustained release using the lipid nanoparticles. And apart from that, we can also decrease the irritation of the drug to the skin by using the lipid nanoparticles. And we have discussed that we are using here the lipid nanoparticles, so they are completely safe and they are biocompatible in nature. So there is no issue, there is no regulatory issue with the lipid nanoparticles, and it has the potential for drug targeting as well. Next slide, please. So these are the various factors which affect the penetration of lipid nanoparticles. So as we have discussed that we are exploring here the lipid nanoparticles. So in case of lipid nanoparticles, the first factor is the size and charge. So if the particle size is less than 100 nanometer, then it can easily penetrate the skin. And can can depot the drug at the dermis level. Apart from that, it also offers the transfollicular route. For the penetration of the drug at the target site. And in this way, it also improves the permission efficiency. Apart from that, if our lipid nanoparticle is positively charged, then because of the negative charge on the skin cells, then it can easily increase the permission efficiency. So in this publication, we have explored the chemistry of lipid nanoparticles, how different functional groups can offer better permeation efficiency as compared to as well as the chain length. Next slide, please. So these are the various challenges in the scalability of lipid nanoparticles. We will also discuss that. First is the reproducibility. As we are using the lipid nanoparticles or any nanoparticles, there is the less chances of reproducibility of the size. So to increase the reproducibility, we should use QBD, quality by design methodology, currently we are also exploring the artificial neural network technique for reproducibility of the lipid nanoparticles or any kind of lipid nanoparticles. Apart from that, we also face the stability issues. So definitely orchestration dynamics. So to decrease the chances of crystallization, we can also go for the that is the NLC, which is the next generation lipid nanoparticles. Next slide, please. So these are the industry scale-up techniques. That is the hot melt extruder. This instrument is currently available in APIIR Hyderabad. So if you want to use this instrument, then you are most welcome. So hot melt extrusion technique offers scalability of lipid nanoparticles. So in this technique, we generally mix the lipid and our drug, and after that, when we pour this mixture into the lipid extruder, then it melts the lipid part as a core to a higher temperature, more than 5 degrees Celsius of the melting point, and then it produces the uniform mixing. Next slide, please. Apart from that, nanospray drying as well as microfluidization technique can also be explored for the commercial scale-up of lipid nanoparticles. So microfluidizer is also available in APIIR Hyderabad. So if you want to explore this instrument for your research work, then you are most welcome. Next slide, please. So this is the rationale of my work. So osteoarthritis, we know that osteoarthritis is the cartilage degradation of the joint, in which after that pro-inflammatory markers are upregulated. For example, TNF alpha, interleukins, which are the pro-inflammatory cytokines, and because of this, it further causes the degradation of the cartilage and produces the joint pain and stiffness. Next slide, please. So in this case, in current research work, we have explored the lipid nanoparticles for the delivery of meloxicam, which is a COX-2 inhibitor. So COX-2 has the ability to prevent the joint cartilage, to prevent the degradation of joint cartilage, as well as being a COX-2 inhibitor, it also inhibits the synthesis or production of pro-inflammatory cytokines like TNF alpha and interleukin 1 beta. And to explore the permission efficiency, we have used different kinds of lipids having the different chain lengths, C16 to C18, and having the different functional groups like amine group, alcohol group, carboxylic acid group. Next slide, please. So these are the basic structures of the lipids which we have explored in this current investigation. So we use palmitic acid, stearic acid, arachidonic acid, behenic acid, having the chain length C16, C18, C20, and C22. Apart from the lipids with different functional groups like amine, stearyl alcohol, stearic acid, and tristearyl. Next slide. So, for the better optimization, we have also explored the in silico docking technique in the current investigation, and we have carried out this work in the collaboration. Next slide, please. So first of all, to screen the lipids, we have carried out the solubility study by using the saturation solubility method. And HPLC was used to analyze to detect the amount of the drug present in the different lipids. Next slide, please. So for the screening of surfactants are especially useful to stabilize the lipid nanoparticles. So we have used hydrophilic surfactants like 2-in-20, 80, F1680, pluronic, as well as so that we can produce the particle size below the 100 nanometer with a stable size. So first of all, lipid and the drug was dissolved in a suitable solvent. And after that, we have carried out the homogenization. Followed by homogenization, we have also optimized the sonication time to reduce the particle size. And followed by, we have screened the surfactant concentration. Next slide, please. So this is the preparation method of solid lipid nanoparticles at the lab scale. So we have dissolved the meloxicam as well as our lipid in a ethanol chloroform mixture. After that, we have sonicated and followed by homogenization, and then we have reduced the particle size. Next slide, please. So in this case, we have fabricated the lipid nanoparticles of different chain lengths with different concentrations of the lipid concentration. and the surfactant concentration. Next slide, please. After preparing the lipid nanoparticles at the lab scale, we have characterized these lipid nanoparticles. So first of all, the first parameter was particle size and zeta potential determination. So if you want to use zeta sizer for your research work, then you are most welcome in APIIR Hyderabad. It is available there. So we have used the dynamic light scattering technique for the measurement of particle size. After that, we have detected the amount of drug which is entrapped in the lipid nanoparticles, and followed by transmission electron microscopy. So transmission, by using the transmission electron microscopy, we have determined the shape of the lipid nanoparticles and the surface topography. Next slide. So the next parameter was the determination of the assay of the drug in the lipid nanoparticles, as well as ex vivo skin permeation study. So for ex vivo skin permeation study, we have used the Frank diffusion cell as per the recommendations, and we have detected the amount at different time intervals. So here we have used the rat skin. Next slide, please. So after preparing the lipid nanoparticles, because our formulation was the topical in nature, so we have entrapped those optimized lipid nanoparticles into the gel, and then we have carried out the characterization of the meloxicam loaded lipid nanoparticles into the gel. And for that, we have carried out the organoleptic properties, determination of pH, rheology and textural properties, as well as dermal distribution using confocal laser scanning microscope. Next slide, please. So following the characterization, our next step was to analyze the therapeutic efficacy of those lipid nanoparticles loaded gel into the monosodium iodoacetate induced knee osteoarthritis model in rats. So for that, we have compared our formulation with the marketed products like diclofenac marketed gel. It is also available as the boviran gel, and etoricoxib marketed gel, and as well as the placebo, that is the blank SLN gel. Next slide, please. So we have used different parameters to measure the therapeutic efficacy, of our formulation against monosodium iodoacetate induced knee osteoarthritis model. In terms of, that is the thickness of the knee joint. So here we have used vernier caliper to measure the thickness of the knee joint, followed by hyperalgesia. So we have used a hot plate method, and followed by motor incoordination test. Next slide, please. So the important part was the X-ray analysis. So we have carried out the X-ray analysis of the rats joints, so that we can image out that is what happened after the application of our topical formulation of meloxicam loaded solid lipid nanoparticle gel. And after that, we have also carried out the histopathology analysis using the toluidine blue staining dye. Next slide, please. Another important parameters in the analysis of the therapeutic efficacy of the gel was the ELISA testing. So ELISA was used to determine the concentration of pro-inflammatory cytokines. That what happened with the pro-inflammatory cytokines when we have applied our formulated gel. So we have carried out all these parameters. Now we will discuss the results. So first of all, we have carried out the in silico docking. So why in silico docking is an important parameter in the optimization of the lipid nanoparticles, because by carrying out the in silico docking, we can preserve our time, we can save our time in the experimental part. So by using the in silico docking, we have seen that, so we have taken ceramide here. For your information, ceramide is an important lipid which is available in our skin, which acts as a barrier in our skin for the permeation of the drug. So those lipids which will show good interaction, with the ceramides, they can be further chosen as the preparation of lipid nanoparticles. so that they can show good permission efficiency in your experimental part. Next slide. So this is the data of in silico docking results. So in silico docking results, we have seen that we obtained a good score with the sterylamine as well as the followed by the steryl alcohol. So good docking score with the sterylamine is indicates that we can pick up sterylamine as a lipid to formulate the solid lipid nanoparticles of your meloxicam drug. So we could not choose other lipids because they are showing the negligible or less amount of that is the less number of the glide score. Next slide, please. So after that, we have carried out the solubility study as we had discussed earlier. So in case of solubility study, we also obtained the good solubility results. That is the consistent to our in silico docking score. So we have obtained the good solubility in sterylamine. So that's why we have selected sterylamine as a lipid core to formulate the solid lipid nanoparticles of the meloxicam. Next slide, please. So in screening of the surfactant, so tween 80 was chosen as the surfactant because it shows the stable particle size with the solid lipid nanoparticles of the sterylamine with the lower PDI below 0.3, which is the recommended value. Next slide, please. So we also optimized the homogenization speed. So at particular homogenization speed, we can obtain the particle size below 100 nanometer or around the 100 nanometer, so that it could show the good permission efficiency when you will apply your formulation over the skin. Next slide, please. So we also optimize the sonication time, because if we would not optimize, then it may cause the crystallization of your lipid. So optimization of sonication time is also important for the preparation of solid lipid nanoparticles. Next slide, please. So in this way, we have carried out the optimization of surfactants of which surfactant or what would be the concentration of the tween 80, which can be used to explore the solid lipid nanoparticles. So at the 2.5% concentration, we obtained the good drug loading capacity 9.1. Next slide, please. So after that, we have fabricated the solid lipid nanoparticles of sterylamine and then characterized for the particle size as we have discussed earlier. So we obtained the size around 217.8 nanometer as compared to the other particles, but still the size was higher than 200 nanometer or 100 nanometer, but still we have chosen this size because it shows the zeta potential of 25 millivolt, which is showing the cationic nature. So we need the cationic nature to increase the permission efficiency. Next slide, please. So this is the data of particle size analyzer. Next slide, please. So this is the surface topography analysis by transmission electron microscope. So it is clearly indicating that whatever we have made in our lab, all the nanoparticles were spherical in shape and they are showing the good strength. Next slide, please. So after that, we obtained the intermission efficiency and drug loading capacity. So sterylamine shows the 91.38% intermission efficiency with the drug loading of 9.14%, which is the good indication for the formulation of our lipid nanoparticles. So after that, we have chosen 3% chitosan, which is also recommended in the regulatory guidelines, that we can explore chitosan as a biopolymer to formulate the topical formulations. So we have chosen 3% chitosan here, and chitosan also possesses the positive charge because of the presence of amine group. And after that, we have, next slide, please. After that, we have carried out the ex vivo skin permission study using the French diffusion cell apparatus. And as indicated, it shows the flux value of which can be measured as microgram per centimeter square per hour. And strialamine has showed 0.76, which was higher as compared to other lipid nanoparticles. So if we see the overall results, then stearyl alcohol, though it disclosed the highest docking score, but still we have chosen the strialamine, which shows the good permission efficiency as well as good flux value in the permission in the Frank diffusion cell operators, as well as we have chosen the lipid nanoparticles of strialamine to formulate the gel, which showed the slow release over the time period till 6 hours. And if you see the results of the based on the results of the docking score, then C18 carbon chain length has shown the highest permission efficiency. So it shows the parabolic relation. So if you will decrease the carbon chain length, then it would not show good permission efficiency. It will increase the chain length. Still, it would not show good permission efficiency, but it showed the parabolic relation. Next slide, please. So after that, as we have discussed, we have carried out the characterization of our meloxicam gel. So we have characterized it for the organoleptic properties as well as the pH value. So the pH was found around 5 to 6, which is recommended for the topical products. Next slide, please. So this is the rheology parameters. So it shows the shear strength, that is the viscosity was decreased as the shear stress was increased. Next slide, please. So here we also measure the texture properties, that is the spreadability. So as it's showing the work of shear, so or the gel strength, firmness. So higher will be the firmness, means your formation is spreadable over the skin. You can easily spread over the skin. Next slide. So we have also carried out several studies for 3 months, and it shows the consistent results as we have seen in that is the assay. The assay value was not decreased over the time period. So we have carried out this image is from the confocal laser scanning microscopy. So we used the cumarin 6 dye. Cumarin 6 dye is a lipophilic dye, so we can use it for that is the permission efficiency, to analyze the permission efficiency. So if we see in the last, that is in this picture, so it is showing the intensity of dye at the lower level. So it is showing the intensity of the dye at the dermis layer as compared to your only gel. So it is indicating that presence of the lipid nanoparticles in the meloxicam gel have increased the permission efficiency up to the dermis layer. And dermis layer also express the COX-2 enzyme. So it means whenever we will apply our formulation over the skin, then it will reach to the COX-2 enzyme, which is expressed in the dermis layer as well as in the joint space. Next slide, please. So this is the measurement of the thickness. In the measurement of the thickness, we did not get the good results because of the autocorrection, because in the monosodium iodoacetate induced model, generally it retreats itself after a few days, so we get not good results. But in case of thermal hyperalgesia, the power withdrawal latency was become normal after a few days after application of the gel. Next slide, please. Same with the motor coordination test. So this is the important parameter as we have discussed earlier, that is the X-ray analysis of the gel. Here we get the good results. So in case of normal control, if you see, this is the gap between this joint space. There is the gap between the joint space, which is showing the good indication for the movement of the joints. So in case of positive control, in which we have induced the osteoarthritis, new osteoarthritis using monosodium iodoacetate, here the joint space was decreased. Here the joint space was decreased. It means you have induced the disease. After that, we have given the meloxicam through oral route of administration, meloxicam through intravenous route of administration, meloxicam loaded solid pin nanoparticle gel toricoxib gel, diclofenac gel, and blank gel. So our meloxicam gel has decreased the has our meloxicam gel has regained the shape of this joint space as compared to the diclofenac gel as well as the blank gel, which showing that our formulation is working enough for restoring the normal condition. Next slide. So we also gained the good results in the ELISA detection. So both the TNF alpha and interleukin 1 beta value was decreased after application of the meloxicam gel. Next slide, please. Same thing with the interleukin 6. So IL-10 is an anti-inflammatory cytokine. So after application of the meloxicam gel, and IL-10 will be increased. Next slide. So another important parameter was the histopathology. So if you see the slide, then the normal control is showing the blue staining. Blue staining because of the staining of the cartilage with toluidine blue dye. And after that, when we have used the positive control, then the blue staining was missing. Here there is no blue staining. It means you have lost the cartilage. We have lost the cartilage here. But in case of meloxicam oral, meloxicam intravenous, meloxicam applied gel, we regain the blue staining, which indicated that now your cartilage is protected. So that is the beauty of our research work. So whereas in case of toricoxib gel and diclofenac gel, we did not get restaining of the cartilage. It means our formulation was better than toricoxib gel or diclofenac gel in attenuating the osteoarthritis inflammation. Next. So this is the osteoarthritis search international. So this is also based on the histopathology analysis. So the osteoarthritis score was decreased and it was comparable to the normal control group. Next slide, please. So conclusion. So this is the publication in the Impact Factor 5, Journal of Drug Delivery Science and Technology after this research work. So if there is any question, then you are most welcome. Sir, we have used the monosodium iodoacetate induced knee osteoarthritis model. Apart from that, we can also use surgery model, but it needs a lot of the expertise.
So in your histopathology, you had shared that, can you just, so have you also tried oral delivery of other agents? Yeah, that's.
Next. Next.
So you have used etocoxib and diclofenac gel, right?
Yes, sir.
Have you also tried the oral delivery of these agents and seen if it is recovering? It is just another question, like not really related here.
No, we have not seen that, but we
Okay.
have taken the commercial formulations.
Right,
So
okay.
both are the topical formulations. So we are compared.
Yes. So in the topical, yours is better. That is very obvious from
yes,
here.
sir, yes, sir.
But in oral, is it also recovering? Like suppose if a patient is using these medicines, or because in the first two panels are oral, right? For meloxicam oral, and the second was is meloxicam IV, right?
Actually, there are the few side effects which are associated with the said
Oral.
administration, as we are reading in the pharmacology. So that's where we explore the topical route instead of the oral route to deliver the meloxicam and the osteoarthritis. Uh, 3 to 5 days. Yes.
If there are no any questions, then thank you, sir.
Thank you very much.
Thank you, sir, for sharing your valuable perspectives. Your session was very informative. Here we end our morning session for the second day of international conference. I request everyone to proceed to dining hall for lunch. I request everyone to gather in seminar hall at sharp 2 p.m.
Come immediately after the lunch. And also you have to fill the feedback form. The link is provided in the WhatsApp group. Thank you.
today's session speaker, finance, our sessions to please guide out the session.
All right. Good afternoon. It's good afternoon in the Philippines. I think it's still good afternoon in India, right?
Good
Can you
afternoon.
hear me clearly? Can you hear me?
Yes.
Okay. I cannot share my deck. Can you make me a co-host? Okay. All right. So I hope everyone's all well. Let me know if my slides are already visible. Can you see it now? Are my
Yes,
slides
ma'am.
visible? All right.
Yes.
So good afternoon, everyone. So again, I'm Felani Royeras Cristobal from the Philippines. So I'm very excited to be with you today. When I was invited and I heard that it's a pharmacy college, I really immediately said yes because my mother is a pharmacist. So your course is actually very close to my heart. So the topic that we will have today that I'd like to share with you is where science meets patients. So it's basically I'd like to just briefly share with you for some perspective expansion in terms of pharmaceutical commercialization. So just a quick disclaimer, any statements, evaluations contained in this presentation does not represent any organizations that I'm associated with. So again, thank you so much for the invitation. So today, my objective is we will be changing perspective. And at the end of this sharing, my objective is for you to feel very proud of what you do for the patients. So I'd like to share with you the story of a bricklayer. So there's this one person, he was asked, person one, what are you doing? I'm layering bricks. So that's the first person said. The second person, I'm actually building a wall. But they're actually doing the same thing, right? And then comes the third person. I'm actually building a very high technology laboratory. So as you can see, the three people are doing exactly the same thing. The difference is just the perspective. So this is what we're going to do today, that outside the realms of science, you're actually doing much, much the world. So maybe the question is, are the scientists and commercial executives like me, are we on the same mission? So commercial executives can probably say, oh yes, I'm marketing products. A regular, maybe a more advanced pharmaceutical commercial executive will say, oh, I'm helping patients achieve treatment success. But in reality, our mission, the scientists, you guys, which in the future, definitely you will be part of that group, is to ensure that the science that you're working on will reach the intended patients and discoveries are fueled further through sustainable commercialization. So as you can see, we're just at the different sides of the coin, but we're on the same coin. So the question, are scientists and commercial executives are on the same side? Definitely we're doing different things, but we're on the same mission. And that one goal is patient care. So I want you to be very proud of the field that you're in. Because maybe I'd like to share with you, when you're already in your respective fields, maybe some of you will discover medicines, maybe some of you will join the pharmaceutical giants in your country. But this is to give you the assurance that commercial people in the pharmaceutical industry will definitely give justice to the efforts of the scientists. So how do we do that? So just to give you an idea how the discoveries are being brought to the market. Launch excellence is one of the things that we should be good at because this is our way of making sure that the molecules will really reach the patients. So launch excellence. So what are the things that usually pharmaceutical companies do to make sure that your discoveries will reach the patients? Market readiness. So we look at the patient journey. Those are the things that we study. Because maybe you're wondering, how do they actually market? How do they know where the patients are? So we actually do patient journey mapping. So if we study medicines for cancer, we really understand where can we catch patients with cancer. If we want to reach to patients with diabetes, we want to understand where are they diagnosed, where do they go to, and those are the leverage points that we're actually planning so that we can meet those patients. Establishing unmet needs. Sometimes there are new molecule discoveries wherein the patients and the doctors doesn't realize yet that they need that. But people in the pharmaceutical industry and you guys in the future will definitely discover some molecules that everyone doesn't realize that they need. Thought leadership preparation. So, of course, we need people who will be advocates of our molecules. Organization readiness. We need to define the go to market strategy. Are we going to do this via virtual, via digital marketing, or via a medical representative route? So, there are actually varying go to market strategy in the pharmaceutical industry right now. So, the field force planning, the cross functional team readiness, and where we're going to sell those medications or the channel planning. So these things are not exhaustive, but these are the things that we're looking into every time we market a specific pharmaceutical molecule. So right now, just to give you an idea, and I'm sure a lot of you will be joining the pharmaceutical industry in the future once you finish your course, is that the pharmaceutical industry is fastly evolving. And these are the changes that happened past 5 years in the industry. So the workforce changes. There are new work expectations right now, especially when COVID happened. A lot of new workforce already requires work from home, virtual work. So what did that do to the industry? We have to explore now new modalities in promoting our medicines. So global factors such as geopolitical considerations, the rising inflation, the supply chain disruption. So really in the pharmaceutical industry, the pressure to innovate gets heavier and heavier. New development, advances in digital technology and user willingness. So now the patients are not just in the clinics, but they're also found everywhere else because a lot of people are now looking into social media, Google, just to study what they're going through, the environmental, social, and governance expectations. So now the pharmaceutical industry, there's now that diffusion of individual players' power. So this is the evolving landscape of the pharmaceutical industry. So if in the future you're joining this industry, you will definitely not be bored because it's really a very exciting industry. So another perspective that we want to challenge, because maybe you're curious, because there are companies who are innovating. They're the ones who are discovering the medications, right? And there are also huge pharmaceutical companies that are marketing branded generics, and we have a lot of that in India, and that's one of the strengths of India. They're really very good in branded generic space as well. So they antagonizing each other because once the innovator brand or drug expires in terms of patent, of course the branded generics comes in, right? So that means they're going to compete in the market share. But are they really competing against each other? So this is the perspective challenge. So maybe some of you will be joining an innovator company. Some of you will be joining a branded generics player. But you have to be very proud on whichever player that you will be joining because both are very necessary. The innovator players are the one who unlocks the science and cater to what the patients need next. But the branded generics also are making sure that we are massively expanding our access to the wider patient pool. So as you see, both of them are very necessary. So this is how the market shape looks like, no? In terms of license type. As you can see, almost half are the originators, but half of it is already branded generics. But this is not the case 20 years ago. It's usually 70% originator and very few branded generics players. But it has evolved over time also because there are more and more innovator molecules that are being available already for, of course, their patents are expiring already. So in a nutshell, this is how recently the market shape looks like in terms of license type. And so the perspective change here is that innovator players and branded generics players are both necessary because the branded generics players will only produce molecules with high relevance. So in the pharmaceutical industry, we always say, if your molecule is copied, that's a status symbol because that means your molecule has a high relevance in the patient's world. So being reproduced by branded generics is a recognition of the scientist's success because that means the need for that molecule is very high. And so these are the trends in innovation in the pharmaceutical marketing. As shared, it has evolved faster, at a faster rate, where productivity is the main consideration. So the omni-channel approach now is very, very high because of course there are technologies coming in. So it's the pharmaceutical industry is evolving with advances in digital health, AI, and biotechnology. The decentralized decision making. 20 years back when I was still in sales, the decision maker is really 80% the doctors. But right now it has evolved because patients now have the power to be involved in the decision making of what medication that I want for myself. And even at the pharmacist level, they also have the power because of the wide range of branded generics available. You can actually also provide options for the patients already. So it's no longer just the doctor deciding for the therapy. So the business model evolution, it has changed a lot already because right now there are virtual medical representatives already. And also right now there are third party promotions, which is part of the trend as well in the pharmaceutical industry. So maybe the question is, so how are we contributing? You know why the reason I accepted this invitation as well? If every time I have global engagements, India is really known to be the pharmacy of the world. And I'm not sure how many of you are aware of that. So just imagine if you join the workforce right after you finish your course. You're being part of those who will cater to the world. But the challenge right now is that we're also facing talent shortages. So there's now a 20% increase in the demand for STEM related courses like yours. So the demand for STEM related roles who will be ready for the workforce has increased significantly over the years. So even the current pool of pharma digital talent is 14% lower than the demand. So that is why kudos to universities like yours who ensures that the talent that will be injected to the workforce will be enough. And as you become part of the pharmaceutical workforce in the future, whether you'll be in commercial or in the discovery part, whatever portion that you will be taking on, you're not just catering to your country, but you're actually catering to the world. So you have to be very proud of the therapy, of the mission that you have for the patients. And so again, the goal of the pharmaceutical industry, whether you're in commercial or you're in the discovery side, is still patient care. So I do hope that when I believe there are more than 400 yesterday, right? So whoever are here right now, once you join fully in the workforce, let's join the battle in ensuring that we really take care of the patients all together. right? And so, with that, I want to say thank you very much, and let me know if you have any questions. Continue touching patients' lives. Thank you very much. Are there any questions? Dr. Manisha? Musin? Am I still audible?
Yes.
Yeah. So do let me know if you have questions with a very brief presentation that I shared.
Hello, ma'am. Yes, section. I would like to summarize about what ma'am has presented. One thing that what ma'am has presented, I would like to say that madam has correlated the relation between innovator and commercial
Branded. Yes.
marketing. Yeah, because how the innovator, when what is the relation between the researcher and patient, this was the first thing that you correlated. And the second thing that how the changes are taking place in pharmaceuticals and what is the important role of a pharmacist. You explained very nicely, ma'am, and I would like to thank you on the behalf of GH Raisoni University for your beautiful, logical, and knowledge enhancing session. We are very thankful to you.
Thank you very much also for the invitation. And I'm really looking forward once you're already, when all of the audience right now are in the workforce. I really wish you all the best as we take care of the patients all together, whether what role we're in. At the end of the day, our role is basically patient care.
Yeah.
Thank you so much, ma'am. Thank you for giving your precious time for this nice presentation. Thank you.
Thank you very
It was
much.
very okay, ma'am. Thank you.
ma'am.
Okay.
Have a nice day.
Have a nice day. Bye.
Have a great day.
Bye.
Karuna, close.
Good afternoon, ladies and gentlemen. On behalf of the organizing committee, we warmly welcome you to this grand valedictory function. I am Shivam Yadav from 7th SEM School of Pharmacy.
And I am Kainat from 3rd SEM School of Pharmacy, G.H. Raisoni University, Saikhera. Today is a day of celebration, reflection, and heartfelt farewells. We have gathered here to mark the culmination of a journey filled with hard work, perseverance, and achievements.
That's right. It's a day to celebrate the accomplishment of our participants, their incredible growth, and the milestone they have achieved. So, without further delay, let us begin today's event.
It is an honor to welcome our chief guest, Dr. Milind Umekar, President of APTI, for today's valedictory function of International Conference on Approach to Pharmaceutical Insight through Interdisciplinary Research 2024. I request Dr. D.C. Sahu, Dean, School of Pharmacy, to welcome Dr. Milind Umekar by presenting a sapling as a token of love and gratitude.
Dr. Milind Omekar sir is President of APTI and Director of Srimati Kishoritai Bhoyer College of Pharmacy, Kamti. Sir has completed his doctorate in Pharmaceutical Sciences and Management. Sir has total experience of 26 years, including academic and research experience. Sir has received project grant of total worth of 111 lakhs. Sir has received many prestigious awards, including Principal of the Year, Ideal Principal, and many more. To his credit, he has attached many national and international conferences in his complete tenure. With such a great profile, I invite Dr. Milind Omekar sir to address the audience.
So, good afternoon to all of you. The convener and the principal of the institute, Dr. Sahu, the speakers of this international conference, Dr. Lad and Dr. Madankar, all the teaching and non-teaching staff of the G.H. Raisoni School of Pharmacy, all the participants, and my dear students. First of all, I congratulate to the G.H. Raisoni group for organizing this international conference on multidisciplinary research. I visited this campus 2-3 times on the occasion of such type of conference, and it is really appreciable that every year GH Raisoni taking efforts to contribute for the research in pharmaceutical sciences. The theme of this conference is on the pharmaceutical insight with interdisciplinary research. As we all are aware that the pharmaceutical research on the drug discovery as well as on the drug delivery systems are continuously going on. And for to speed up this research, we need the support of the other disciplines too. Because only one discipline can't solve the expectation or requirement of the research. And that's why I congratulate to the team for selecting this theme for this international conference, because there are many areas where we require the help of other disciplines. Like nowadays, 3D printing in drug discovery is very famous, as well as nano technology in the drug delivery systems. Such type of techniques will be possible only with the help of the peoples who are expertise in that discipline, like engineering or like computers. And we saw that during COVID, we were able to introduce or discover the Covishield vaccine very fast. That was possible only because of the AI and data science. Because lot of data they compile and they support for the clinical trials of the uh, vaccines, and we get the vaccine very fast within a time. So such type of continuous support from the various discipline is going on in the pharmaceutical sciences. And if we want to fulfill the requirement of the research in pharmaceutical sciences, we have to take the help of other disciplines too. So I hope the speakers of the this 2 days conference already brief you about the new technologies, as well as the about the theme of the conference. And I think all the students take the benefit of this conference to upgrade their knowledge or to know about the research going on in the industry. uh, and I'm also thankful to the management of this institute, because they gave me the opportunity to interact with Madankar sir and Lad sir, and it was a great interaction with them. And I hope all the students of GH Raisoni, as well as the faculties from the various institute, as well as the participants from the various pharmacy college, will take benefit of this conference, and they will try to work hard for working for the research, as well as to create some innovations. As you know, I'm working as a national president for the Association of Pharmaceutical Teachers of India, and being a national president, we are also working for the betterment of the profession as well as pharmacy teachers. And for that purpose, we are continuously working. And I assure in next 3 4 years during my 10 years, we will see the lot of changes in the field. We are continuously in touch with the Ministry of Health, as well as Family Welfare, as well as we are in touch with the Pharmacy Council of India. And because of the regular follow-up from the Association of Pharmaceutical Teachers of India, uh, in last 2 days, PCI declared the GPAT stipend for the GPAT qualified student, which was pending from last 1.5 year. And I'm also thankful to the Pharmacy Council of India for they accepting our request, and their support to the student of pharmacy. Similarly, we are continuously in touch with the pharmacy PCI president for the exit exam for the diploma student. We are in favor of exit exam, but we insist them to conduct only one paper instead of three papers, because Pharmacy Council of India is planning to conduct three papers, and they are going to charge around fees 6,000. So we request them to reduce the fees and also you take only one paper, so that it will be easy for the student with the poor family financial background. And they accepted, and they stopped. They gave the stay to the exit examination. And I hope in next 1 or 2 months they will declare the new notification of the examination of diploma with only one paper and with minimum fee for the examination. That exam will be conducted by the National Testing Agency, and it will be really helpful for the student. Similarly, we are also continuously in touch with the state government as well as central government to stop the mushrooming of the pharmacy college, and to improve the quality of pharmacy students, as well as to improve the quality of pharmacy education. And I hope yesterday only my vice president Dr. Dipendra Singh was here, and he is also working as a chairman for the PCI education regulation team. So I hope they already discuss all the things with the principal, and we are trying continuously for the betterment of profession. On this platform, I would like to share one thing with you. Recently, Association of Pharmaceutical Teachers of India, is declared as a organization member of International Federation, Pharmaceutical Federation, that is FIP. It will be really helpful for the pharmacy teachers to getting the international exposure at various level. And next year, FIP 2025 is going to be organized at Denmark, and it will be the great opportunity for the teachers to interact with the teachers at international level. It will give a platform to the student as well as teachers also. And also we are going to introduce now institutional chapters at every institute, so that the institute as well as teacher can continue the APTI activities or APTI related in association with APTI. They can organize multiple activity in their institute only through the institutional chapters. So once again, I'm thankful to the principal for giving opportunity to interact with you all, as well as giving me opportunity to uh, give the recent updates about the APTI, give the opportunity to share the recent updates of APTI with you all. I assure you all we are trying at our level best for the betterment of student, and we will be definitely give the good education in very good few days, because PISA is come up with a new syllabus, and they are also planning to give the um, opportunity to the individual institute to change the syllabus by 30%. So it will be really helpful for us to introduce some industrial requirement in addition to the PCI syllabus in the our syllabus, so that it will be really helpful for the student to upgrade their knowledge and to make them industry ready. So I congratulate to the entire Raisoni group for organizing this event, and I hope the student will take benefit of such conferences, because मैंने बहुत बार देखा है कि स्टूडेंट कॉन्फ्रेंस में जाके बस चुपचाप बैठे रहते, तो चुपचाप बैठने के लिए आप लोग यहाँ पे बुलाया नहीं जाता. ट्राई टू इंटरेक्ट विद द टीचर्स और ट्राई टू इंटरेक्ट विद द स्पीकर्स टू गेट द रिसेंट ट्रेंड और रिसेंट जो भी अपग्रेडेशन रहता है इंडस्ट्री का, वो भी आपको पता होना जरूरी है. बिकॉज जैसे अभी हम डिस्कशन कर रहे थे कि व्हेनेवर स्टूडेंट एडमिट इन द फर्स्ट ईयर, दे आस्क, पहला क्वेश्चन उनका प्रिंसिपल से यही रहता है, सर आपके कॉलेज का पैकेज कितना है. अभी फार्मेसी में आए भी नहीं, फार्मेसी पी से लिखती है कि एफ से लिखती है ये भी पता नहीं, और पूछते आते हैं कि सर पैकेज कितना रहेगा. तो मैं यहाँ पे क्लियर करना चाहता हूँ, पैकेज नाम की कोई चीज़ नहीं होती है, वो सैलरी होती है. बस ये नॉलेज अपने दिमाग में डालते हैं ट्यूशन क्लासेस वाले 11th-12th के बाद में, कि बहुत बड़े बड़े ख्वाब दिखाते, JEE क्रैक करोगे तो IIT मिलेगा, फिर करोड़ का पैकेज मिलेगा, या फिर नीट पास करने के बाद डॉक्टर बनोगे. लेकिन डॉक्टर 15 साल बाद बनती, ये नहीं बताते कि बिकॉज यू हैव टू गो फॉर MBBS, देन MS, देन सुपर स्पेशलिटी, देन 5 years किसी के अंदर प्रैक्टिस करो, 40 साल के एज में अर्निंग शुरू करो. ये चीज़ें कभी बताते नहीं है, और उसी के शायद आजकल के बच्चों में ऐसा लगता है पैकेज मतलब यानी बस कोई भी डिग्री कॉलेज में एडमिशन ले तो पहले पैकेज पे. पैकेज किसी कंपनी पे या किसी कॉलेज पे डिपेंड है, इट इज डिपेंड ऑन इंडिविजुअल पर्सन. आप किस तरह काम करते हो, आप कैसे काम करते हो, आपका एटीट्यूड कैसा है, आपकी लीडरशिप क्वालिटीज कैसी है, ये सब चीज़ों पे डिपेंड होता है. तो ये 4 साल है ही उसी के लिए, अपना नॉलेज अपग्रेड करने के लिए, अपनी खुद की क्वालिटी, अपनी पर्सनालिटी, अपने लीडरशिप स्किल, अपने सॉफ्ट स्किल सभी आपको डेवलप करना है, ताकि वेन एवर यू आर गोइंग टू फेस द इंटरव्यू, दैट टाइम डेफिनेटली दैट HR पर्सन और टेक्निकल पर्सन विल सिलेक्ट यू. और फिर उसके बाद में उसने पूछना चाहिए कि बेटा कितना लोगे. आपने 3 Idiot में डायलॉग सुना है, यार आखिर बताओ कितना चाहिए. तो वो क्वेश्चन सामने वाले ने आपको पूछना चाहिए. वो कब पॉसिबल होगा, जब आप अपना 100% अपने डेवलपमेंट पे खर्चा करोगे. क्योंकि यहाँ पे इंडस्ट्री वाले भी लोग हैं, यू कैन आस्क देम कि सर आपके बैच में से कितने लोग आज इस पोजीशन पे. वेरी फ्यू, कुछ ही लोग रहते हैं जो वहाँ तक पहुँच पाते हैं. तो यू हैव टू ट्राई अकॉर्डिंगली, यू हैव टू प्लान योर करियर, and I wish all the best to each and every participant. And I also thanks to the speakers for their valuable time, and they share their experiences as well as they share their expertise with all the students. Thank you very much.
Thank you so much, sir. We are deeply grateful for your presence and the wisdom you have shared with us today. Your words will resonate with us as we embark on the next chapter of our lives. Now, I would like to call Ms. Nandini Sahu, Assistant Professor, Sardar Vallabhbhai Patel College of Pharmacy, Balaghat.
Good afternoon, sir. Good afternoon, all. My name is Nandini, and I am from Sardar Patel University, Balaghat. So, so session was very good, and the experience was very good. And thank you. I want to say thanks to Raisoni University and organizing committee for organizing this wonderful conference. Um, that is very informative, and that was very good. Okay, thank you.
Thank you, ma'am. As we move forward into the future, it's important to remember that we have all been given the foundation to succeed. And now it's time to put that knowledge to work. Ladies and gentlemen, as we come to this special segment of our conference, it is time to recognize the exceptional contribution of our participants. Today, we celebrate the dedication, creativity, and hard work that have shown through in the best oral and poster presentation. We begin with the award for the best oral presentation. This recognition goes to an individual who has not only demonstrated outstanding research but has also conveyed their findings with clarity and impact. The recipient of this award is Dr. Deveshri Mandurkar, Balpande College of Pharmacy. Give a big round of applause for our winner. I would like to call upon Dr. Milind Domekar and Dr. Abhijit Munshi to give away the prizes. A big round of applause for a winner. Award for the runner-up of oral presentation of APIIR 2024 goes to Amruta Shetty, Balpande College of Pharmacy. Give a big round of applause. Award for consolation prize for oral presentation goes to Sama Jabas ma'am in online mode. Let's have a big round of applause.
Now, we move on to the Best Poster Presentation Award. This prize acknowledges the innovative approach and visual communication skills that captured the essence of impactful research. I am delighted to announce that the award for the best poster presentation goes to Disha Dabade, Nehru College of Pharmacy. Have a big round of applause. If anyone is there from Kamla Nehru, he or she can come to the stage.
To collect the prize.
Award for the runner-up of poster presentation of APIIR 2024 goes to Raman Jaiswal from KC Bajaj College of Pharmacy.
Give a big round of applause.
Award for Consolation Prize of Poster Presentation goes to Shamal Wankhede, APIIR Wardha. Have a big round of applause. Thank you so much, sir.
Congratulations to all our winners. Your achievement inspire us and set a benchmark for excellence. Thank you everyone for your participation and for making this event a success.
Distinguished guests, respected delegates, and participants, as we approach the conclusion of this remarkable international conference, we have one important gesture to make, an expression of our deep gratitude and respect. The memento symbolizes our heartfelt appreciation for valuable time, knowledge, and support in making this event memorable. I would request Dr. Milind Umekar to present memento and certificate of appreciation to our today's eminent speakers, Dr. Rahul Lad and Dr. Jitendra Madan.
Give a big round of applause. Now, I invite Dr. Meena Rajesh, Vice Chancellor GHRU Saikheda, to kindly present the memento to Chief Guest. Thank you, Dr. Milind Omekar, for your gracious presence. We have deeply honored and we look forward to your continued support and guidance in the future.
As we come to the close of this wonderful ceremony, we would like to invite Dr. Manisha Kavatkar, convener APIIR 2024, to the stage to deliver the vote of thanks.
Good evening, everyone. Thank you, Shivam and Kainat. So, good evening, distinguished guests, esteemed speakers, respected colleagues, Honorable Dr. Milind Omekar, sir, Honorable Vice Chancellor Dr. Meena Rajesh, ma'am, Dr. DC Sao, sir, and Dr. Jignesh Doshi, sir. My warm good evening to one and all here. So, as we draw the curtains on this extraordinary journey that we have shared in last 2 days, my heart is actually filled with immense gratitude and a sense of fulfillment. It is with great pleasure that I stand before you today to express my deepest appreciation to all those who have contributed in making the international conference APIIR such a rebounding success. Actually, there are moments in life when we come together united by a common vision and create something truly special. So today, I stand before you overwhelmed with emotions as I reflect on the tireless efforts, boundless enthusiasm, and collaborative spirit that each one of you has brought to this conference to make a success. So it is because of your unwavering support and collective commitment that we are able to celebrate the success today. We are able to celebrate the success of this event. As we conclude the APIR, it is my privilege to extend heartfelt vote of thanks to everyone who contributed to making this conference a big, what I can say, a big success to GH Raisoni University. So I would like to take this opportunity. First, my deepest gratitude goes to our distinguished speakers and participants for sharing your invaluable insights, your contributions that have inspiring and instrumental in making this conference a grand success. My special thanks to our Honorable Chairman, Shri Sunil Raisoni, sir, our Honorable Executive Director, Shri Shreyas Raisoni, sir, whose overall direction and wisdom have shaped the success of this conference. Your belief in the potential of this event and your continuous support have helped bring this vision to life. First and foremost, I extend my heartfelt thanks to our Honorable and lovable Vice Chancellor Dr. Meena Rajesh, ma'am. For your unwavering support and vision, ma'am, your leadership has been a source of inspiration to all of us, and your commitment to the advancement of research and education has made this event possible. We truly appreciate your continued encouragement in shaping academic initiatives like this conference. I would also like to express my sincere gratitude to Registrar Sir Dr. Sriram Joshi, whose administrative expertise and timely support have been invaluable throughout the planning and execution of the conference. So your behind the scene work ensured that everything proceeded smoothly, and we are deeply thankful to your guidance. I would also like to extend my gratitude to our Chief Finance Officer, Shri Dheeraj Moryani sir, and Account Department, Ms. Deepa and Ms. Saloni, for your exceptional support throughout the entire planning and execution of this conference.
I extend my special thank you to our sponsors, Arco Life Sciences, Deepti Pharmaceuticals, Go Vigyan Anusandhan, Devlapar, and Alchemy Clinical Research, whose generous support has been invaluable in bringing this conference to life. Your commitment and partnership have enabled us to achieve our vision and make this event a memorable experience for all of us. A heartfelt thanks to Dr. Abhijit Munshi sir, Director Alchemy Clinical Research Services, for your generous sponsorship for this conference, sir. Your support has been crucial in making this event a success, and it highlights the vital role of collaboration in advancing pharmaceutical research and interdisciplinary insight. We are especially grateful for you sponsorship of the prizes, which have added great value to this conference. Additionally, we deeply appreciate you. You have spent these 2 days with us by giving your precious time and sharing your expertise and insights. Your presence has truly enriched us with wisdom. I would like to offer my sincere thanks to organizing chair Dr. D.C. Sahu for his outstanding leadership, meticulous planning, and coordination. Your vision and hard work have been instrumental making this conference a remarkable success. Thank you, sir. A big thank you to our co-organizing chair, Dr. Jignesh Doshi, sir, for dedication, attention to detail, collaborative spirit you brought to this event. Your constant support and active involvement ensured that every task was executed seamlessly throughout the conference. I would also like to acknowledge the incredible efforts of each committee member that worked tirelessly behind the success. A special thank you to co-conveners, Professor Amol There and Professor Ganesh Derkar, for their leadership and dedication in planning and executing the event. Our registration desk, Professor Tabassum Bano, along with Tabassum ma'am, Professor Manish Baheti, Professor Satish Bodle, Professor Sapna Pal, Professor Munal Ade, managed online and offline registrations smoothly and ensured a seamless process of all attendees. A big thank you to the Registration Kit and Certificate Committee, Ms. Pooja Vaure, with support of Praful Hire, Ms. Sankita Lonkar, Ms. Bhamna Raut, Ms. Shireen Nas for their diligent work in preparing and distributing the registration kits and certificate to participants. The scientific editorial chair led by Mr. Amol There, Dr. Venkatesh Suresh Babu, Dr. Sagar Trivedi, and supported by Ms. Mona Salvatkar, Ms. Abhilasha Yadav, Ms. Chetna Selukar, and Ms. Neelam Nehare. They all manage sessions very well. They organize session very well, online and offline, abstract screening, and ID creation with the precision as very much care. A special mention to the Publicity and Public Relation team, Dr. Dhanashri Tumme, Ms. Deepika Devnani, Mr. Moshin Khan, Mr. Satish Bodle, Mr. Manish Baheti, and Prerna Patil for their excellent work in promoting the conference both online and offline. My heartfelt thanks to Inaugural and Stage Committee led by Ms. Anmol Dhavande and supported by Ms. Akansha Bhandarkar, Ms. Hira Karemure, and Ms. Shikha Prasad. Thank you for managing the stage organizing, mementos, coordinating with awards, abstract, utmost great and professionalism. My heartfelt thanks to Food and Hospitality Committee led by Ms. Deepika Devnani. Hope you all enjoyed the food on both days. Thank you so much. And along with Ms. Deepika, Mr. Sonu Ghate, Mr. Moshin Khan, Dr. Venkatesh Babu, and Ms. Purnima Patil for their meticulous planning and warm hospitality. I thank you very much. Now, our sincere thanks also go to Transportation and Hospitality Committee, which was led by Mr. Ganesh Derkar, with Mr. Moshin Khan, Praful Hire, Ms. Sapna Pal, and Ms. Prerna Patil for ensuring the smooth transportation and accommodation of our guests and participants. Hope you all enjoyed the journey from Nagpur to Saikheda. Okay. So I would like to extend a special thank you to our non-teaching staff, supporting staff, especially Uddhav Bhandarkar sir, for their invaluable support, for your dedication and hard work. I would also like to acknowledge Mr. Sanjay Das, who is our administrative officer, for his efficient management of resources and coordination with all departments. Your attention to the detail and organization skills have been fundamental to the smooth functioning of this conference, sir. This conference would not be possible without your support. Thank you, sir. Special thanks to Ashish Mohatkar, sir, from store department, whose logistical support ensured that all the materials required for the conference were available and organized on time. Your contribution has made everything seamlessly. Hope sir is there. Thank you, sir. A special thank you to IT support team, Ashish Gadbande sir, Sarang sir, along with public relation team, which is led by Yogesh Trivedi sir, who although come from JHREC for setting up here YouTube live streaming and all. And your dedication and innovative approach ensured us conference message reached far and wide, gathering enthusiasm and with engagement of the participants. So lastly, my deepest gratitude and thanks to my beloved students, your hard work from designing the selfie corner, setting up the stage with full enthusiasm by night 11 p.m. or 12 p.m. and calling that, madam, everything was ready. You can get surprise in tomorrow morning. So thank you so much, dear students. You brought an engaging, vibrant touch to the event, and for that we are very truly grateful to you, we all organizing committee member. So to all our attendees, both online and offline, thank you for your enthusiasm and engagement. Your active participation has energized and inspired us all. On behalf of the entire organizing team, I extend my profound gratitude to each and every one of you for your contributions, guidance, and support, which have made this conference an inspiring and successful event. We look forward to more collaboration and hope to see you all at our next conference. Finally, a heartfelt thank to everyone who contributed, whether directly or indirectly, to making this conference a resounding success. Your efforts, seen and unseen, have truly made a difference, and we are deeply grateful to you from GH Raisoni University, Saikheda. So, as I announce each name, let us now move into the felicitation of our organizing committee members who tirelessly work from last 3 to 4 months to making this conference a successful event. So I announce each name and kindly request Dr. Milind Umekar sir and Dr. Meena Rajesh ma'am to felicitate our committee members. First, I request Professor Amor Tere sir, who is co-convener of the committee, conference. Hello. Professor Ganesh Derkar. Our registration chair, Professor Tabassum Bano. Professor Manish Baheti. Professor Satish Bodle. Miss Sapna Pal. Miss Sapna Pal. Miss Mrunal Ade. Your smooth handling of the registration process, both online and offline. Now, registration kit and certificate committee, Miss Pooja Vaure. Miss Ankita Lonkar. Miss Bhavna Raut. Bhavna is nothing but a backbone of the registration committee. She handled registration very well. Please give a big round of applause. Miss Shireen Naz. Miss Sapna Pal. Now next is inaugural and stage committee. Inaugural and stage committee led by Miss Anmol Dhavande. Miss Akansha Bhandarkar. Miss Hira Karemure. Miss Shikha Prasad. Scientific editorial team, Dr. Venkatesh Suresh Babu. Miss Mona Salvatkar. Miss Abhilasha Yadav. Miss Chetna Selukar. Miss Neelam Nehare. Publicity team, Dr. Dhanashri Tume. Mr. Mohsin Khan. Mr. Praful Hire. Food and Hospitality Committee, Ms. Deepika Devnani. Ms. Purnima Patil. Mr. Sonu Ghate. लगता है सोनू सर के फैन ज्यादा है, सर ने खाना अच्छे से खिलाया है सबको। अब मिस प्रेरणा पाटिल। I request Sanjay Das sir. Sir is tirelessly working for us from last 2 months. I request Ashish Motkar sir. I request Yogesh Trivedi sir. Navneet Suryavanshi sir. Ashish Gadbande sir and Sarang sir is not here. So thank you so much everyone for giving your precious time. Thank you so much sir and ma'am for felicitating our organizing committee members. A big thank you to my beloved students, Sopan Gupta, Sania Sheikh, Kainath and Shivam Yadav for outstanding anchoring and comparing from 2 days. Thank you so much all for being here with us for 2 days. Thank you so much. Please gather here for group photograph. पर मुझे लगता है यहाँ आएगी नहीं. नीचे हम जो रजिस्ट्रेशन काउंटर है वहां पे स्टेप्स पे ग्रुप फोटोग्राफ ले सकते हैं अच्छे से. सो प्लीज गैदर एट स्टेप्स ऑफ ग्राउंड फ्लोर.